we have a real treat for you, and those of you that are in here on time, like high five to you, because this is Dr. Brenda Porter, one of my favorite people in the world. She is a pediatric epileptologist at Stanford. Do you see adults too? No, she doesn't see adults too. And she is very, very knowledgeable and very wonderful. And we just heard a great talk from Dr. Knupp about finding the cause. What is your loved one's LGS secondary to? And so Dr. Porter is going to spend a lot of time talking about genetic cases and acquired cases of LGS and how you can find your cause. She has also offered to spend time with families today with one-on-one counseling. You would sign up at the registration desk. She's meeting with families today from 1 to 4. So if you want to sign up, make sure you do that, and you can spend some time talking to Dr. Porter personally about your case and finding your cause. And with that, I'm going to turn it over to Dr. Porter. Okay. And yes, mask off. Great. Thank you.
Thanks, everyone. Yeah, basically, I did this last in person in Seattle. I think it was Seattle. Was it Seattle the last time you had this in person? Yeah, okay. So I did this, and several other people did, and it was interesting. People just kind of came in and talked about different things. It's not like there's a specific agenda, so you could talk about anything. Okay, so I'm going to sort of think about it in the relationship of why does your loved one have LGS, but then also, like, what does that mean for your treatment plan? Because I think just understanding the cause without understanding that it has implications for the therapy isn't that helpful. I also designed this talk a little bit to think of it as, so you're my patient and I've just given you a diagnosis or given you a possible diagnosis. What are the things that I don't have time to talk about with you at the visit, always? Okay. So we'll do a little bit of background on genetics. I'll talk a little bit about laboratory studies. Then I'll go into some detail about imaging and how now, anyway, imaging often meets genetics. And a lot of things that we still don't know. So some of your loved ones won't have a diagnosis still after we do all these things. But there are things you can do to make it more likely you will get a diagnosis, quickly if something new comes along. And then I'm going to talk a little bit about the future. Okay, so when we think about genetics, I like to think about it as you're going from the big to the tiny. And the big, in this case, are chromosomes. We all have them. They're these big structures that look like an X. And sometimes you can have too many. The most common thing is called trisomy 21. So instead of having 2 21 chromosomes, you have 3, and then you have something called Down syndrome. And then you start to go a little bit deeper, and you start to look for pieces that are missing or extra. So instead of having all of chromosome 21, maybe you have part of it, and a little bit's missing. And that's analyzed through a test called a SNP, or a microarray. The next thing that I think of is, let's think of all the genes that have been identified that cause Lennox-Gastaut or severe epilepsy. They've put those together in what's called a panel, and they look at those genes. There's 300, 400 genes in these panels. Many companies have them. And that's probably the first step, because those are genes that are really commonly associated with epilepsy. And then you go a little bit deeper and you look at the 30,000 genes that are in that genome, and you look at each gene and you sequence it. And that's called an exome. And that really looks at the parts of the DNA that make a protein. So those are the things that make your cells work. But there is even another layer after this exome, where you have all the other parts of the genome that don't make a protein, but maybe tell that protein to be made. So they're kind of the control elements for the actual production of proteins. And those you would only get to with a genome. So I think that when I think about it, I always do a gene panel if I don't have a diagnosis. They're free. Well, they're mostly free. And they'll get a lot of people diagnosed. The next step is the whole exome. There it becomes more important to think about who's ordering the test, because you get a lot of data that doesn't always make sense. So I asked my genetic colleagues, I have some neurogenetics colleagues, to order those, to meet with you, to explain what they're doing, because the parents, or the biologic parents, have to give a sample as well. And you can find some things you weren't expecting to find. So it's a little bit more complicated, and they have the expertise to give you advice on what they find. And then right now, what I was just told not, genomes tend to be research studies. And there, for sure, I would ask one of my genetic colleagues to do it. I do have quite a few patients that have had them because Stanford has them in their lab being done, but the analysis is very challenging. And you definitely need to have a comparison between the parent and the affected child. Okay, so I want to go through some terminologies, because many of you may have had genetic testing and you end up with this thing called a VUS, which is a variant of unknown significance. And that really means that there's a difference in the way the sequence of that gene is compared to most people in the world. It doesn't mean that it's actually causing any problems, and it may not be causing any problems at all, and that's why it's of unknown significance. I would say maybe half the studies I do where I get an exome, it has one of these. And it's frustrating because people will think, oh, well, this isn't good. But sometimes it's just a difference. We all have them. But the question is it causing a problem? And that's tougher to know often. Another term that comes up frequently is called de novo. And de novo means that the patient has it, but the parent doesn't. And that's a new change, because if they inherited it from the parent, then it would be in the parent's genome. So de novo means new. And then I realize these slides are not the most recent ones, but anyway. So then another thing that happens with that whole set of data you get, they compare the results to what's known in the literature. So they'll take the patient's results and they'll compare it, that sequence, and they'll say, oh, we know that that's caused problems in another patient, so that's probably causing the disease. But they may also see it in the literature and be like, oh, this caused no problems in this patient population, so we know that that's not an issue. And I think this is important. If you have your neurologist explaining it to you, like I just did, we're not experts at this, right? I learned how to read EEGs and all these other things, but there are doctors that really specialize in this, and they're called genetics doctors. And now we train them often to be neurogenetics doctors. So they finish their neurology training, and then they go on to do a genetics fellowship. So if you're not understanding what your doctor is telling them, ask them to refer you to somebody that really is a world's expert in genetic diagnoses. I just mentioned this VUS, and it can be very frustrating. If it's a VUS that the parents don't have, so it's something de novo and a VUS, that makes it more likely it might be pathologic. Sometimes you need just more time and data. You know, every day new information is being put out there about genetics. So just time may give you your diagnosis. There are fancy ways to test VUSs. So some of my patients will have the protein that's supposed to be made actually studied, as opposed to just the gene. That can be a very helpful way. If that protein's not being made, then something's wrong. So we can go to a little bit farther along and really figure out whether the VUS is causing a problem. And then if you don't have a diagnosis, go back again and again and again, because oftentimes every 6 months to a year, something new may pop up in your test. Most companies now will do that for you. They will go back and look at the data, but if it's really an old study, like your exome was done at Baylor 10 years ago, it may not be analyzed repeatedly. So go back and ask again for help. I always see in the genetics note, come back in a year. And many patients don't come back in a year because that appointment wasn't set up, and they don't realize that they really need to possibly go back to get a diagnosis. Okay, do laboratory studies, and when I say this, I don't mean genetic testing, I mean like a blood sample for some metabolic test. Are those still important? And I think they are if you don't have a diagnosis from the genetic testing, or it's unclear if you have a diagnosis from the genetic testing. But we don't do them as often because I don't think in the age of genetics it's as important. They can confirm a diagnosis, or they may point to a diagnosis. But the way to probably do them now is to be a little bit fancier. So in the past, you would basically just tell the lab, I want paradoxical 5-phosphate measured or something like that. We don't do that anymore. Now we do these things called metabolomics, which is B on this slide. And it looks really complicated because it really is. But each one of those little dots is a sample, a measurement from one sample. So this patient gave blood, and they measured 1,000 different things in the blood. And the red and the blue, bigger dots, are the abnormalities. And they can look at that, and they can see that these 10 things are up, and these 5 things are down, and that's maple syrup urine disease. Same thing with phenylketonuria, those red dots, that's abnormal. So you don't have to go specifically and say, I want to measure this and this to the lab. Now they can do metabolomics and just measure it all. And that can pick up a few diagnoses. I've had that happen where there was like very specific pattern, and that was the diagnosis. It's especially important if I do a seizure panel, I do an epilepsy panel, and yet it turns out the patient has something that isn't in the panel, but is picked up this way. So imaging is really the way we've always diagnosed children with neurologic diseases. But I think genetics now may play an even bigger role. So do not think imaging alone. Think imaging and genetics. It still provides the answer for quite a few patients, but it's also important to realize that just like you have a VUS in the genes, you can have VUSs in MRIs. So when I say that, I just mean that often MRIs can be slightly abnormal. It's not uncommon to have a reading with this sort of, increased Virchow-Robin spaces or something very commonly found that is not contributing to your child's disease. And it really, you need to put the MRI into context of the disorder. And so if things don't match up, it's your neurologist's role to say, well, it's abnormal, but it's not very abnormal, nor would it explain this disorder. And then I think the most important part about getting good imaging, and Kelly just mentioned this, you really need a good image, because some patients are probably now eligible for surgery that would not have been eligible in the past. So possibly if you've had a 1.5 Tesla MRI 10 years ago, you need a 3 Tesla, because there might be some surgical approaches that hadn't been really thought to work in the past. So this is one of the most common causes of diffuse disease that leads to Lennox-Gastaut Syndrome. So this is a picture of hypoxic ischemic encephalopathy. So these are babies that are born and have not received enough blood supply or oxygen to the brain right around birth. And we look for this, and it tells us that there is kind of a diffuse injury. This patient probably would not benefit from surgery, but might benefit from certain medication trials, or I'll talk a little bit on Sunday about DBS and RNS as possibly a therapy for these more diffuse injuries. But also babies can be born and have a focal injury. This is a child with a stroke around the time of birth. And they often will have a weakness on one side. They can have Lennox-Gastaut, and yet really they would benefit from having that, what's left of that hemisphere disconnected. So they would be a surgery candidate. So just having early life injury is very dependent on what the injury was. And then imaging meets genetics. So these are all patients that I would think highly likely to have Lennox-Gastaut syndrome. And that would now be found on the genetic testing, but the MRIs have certain characteristic features. I'm sure I can make a point. No, okay. Well, as you go across, there's sort of, what you see is that some of the MRIs have very smooth surfaces. That's called lissencephaly. And as you go across, there's different grades of lissencephaly and different genetic causes. And these are also patients that I would not do resective surgery on, but I might place a DBS in, perhaps. So this gives us genetics and the imaging match. So we know that these patients have certain genetic causes, and then there are certain therapies that might be better based on the literature. These changes are usually found throughout the body, so, and they are primarily de novo, so the parents would not carry them. Okay, so, oops, that's a little weird there. Okay, so yeah, sorry, these are a little bit out of order, but we'll go ahead and do this slide. So when you think about what it means to have a genetic diagnosis, it can be very helpful because there's this term precision medicine that people use, and what it really means is we take all patients with Lennox-Gastaut, all of them, and we put them in a study. For instance, the way the Epidiolex trial was run, or the fenfluramine trial, this is how it was done. All patients had a Lennox-Gastaut syndrome, and they all got the treatment. But maybe the way we ought to think about this is, let's say your child has a mutation in a specific gene, and they are blue on this panel. They may benefit more from treatment A than treatment B. So maybe treatment A would be Epidiolex and treatment B would be fenfluramine. Based on the genetics, there may be a better medicine for your child if they have that specific genetic cause. And I think we're not quite here yet. I mean, you saw all the trials are being done generally in Lennox-Gastaut, but it may be worth picking out what the causes are and then looking at the data more carefully to see if there are specific treatments that are better than others. And there's, of course, she mentioned this avoiding medications. So that has been around for quite a while. There's certain sodium channel mutations, and patients can do poorly on sodium channel drugs. Interestingly, there are sodium channel mutations in other sodium channels, and patients do very well on sodium drugs, so sodium channel inhibitors. So it really comes down to somebody knowing SCN1A, avoid these drugs. SCN8A, use those drugs. So it's very important that the physician that's taking care of you understand these differences, and it's evolving, so you really have to keep up with the literature. And then, of course, you know, the ketogenic diet is extremely helpful in patients that have glucose transporter deficiency. It's almost curative in some of them. So again, genetic cause, treatment. Resective surgery can be an option now, even in patients that have multifocal seizures, and those are often also picked up genetically. However, they're not picked up in the blood, so this is a little bit tricky to think about. But so all these patients on this slide had epilepsy surgery, and they all have different causes for the brain causing seizures. They all had seizures. And the red areas are the resected portions of the brain, and what you can see is some of them have had areas that are very large, some have small areas, some have bilateral areas, and basically The genetic diagnosis was not made until the tissue was sent to the laboratory and was analyzed. These are called somatic mutations, so they're not found in the blood. They're only found in the tissue that goes to the lab. But they do have a genetic cause, and it was discovered at that point. What happens if you have your gene, but precision medicine is not available? So you have a genetic diagnosis, but no one yet has really come up with a therapy. My suggestion is that you go to social media. Almost every gene now has a family Facebook group, I would say, and you link yourself to that group. And there's power in numbers. I was just at the SLC 13A5 meeting in Dallas yesterday, and they had 100 patients, 125 patients they've discovered in the past 5 years, and there were people from virtually as well as in person from around the world. And they have a gene therapy that's probably going to be trialed in maybe late this year, early next year. And that's really only after the disease was discovered 5 years ago. And the whole reason that that's moved so quickly is because the family organization has really banded together to get that gene therapy in place. I have patients who can't find a family group, or I can't find any data on that gene, and I put it in something called Gene Matcher. There are other programs, but what Gene Matcher does is I put in my information, KCN5H or something like that, and then every other doctor that has a patient with a mutation in that gene puts their, well, not every doctor, but many of them will put their information in, and together a paper is coming out on that, because we all got my patient and 2 patients here and 3 patients there, and together we came up with a large enough patient population to really write a paper on that gene. And the only reason that was possible is because we were all able to get together from around the world. And then I would highly recommend, if there is a natural history study for your particular gene, you join that. The natural history studies, I think, are feeding in to the gene therapy studies. They're not only feeding into the gene therapy studies, there are some companies that are working on specific potassium channels drugs. That will be the way to get into that trial earlier, is to be part of the natural history study, because then the researchers that are working on that gene will get you into the medication trial or gene therapy study. And I think there are many ways to fix genetic disorders of the nervous system. They're all complicated, and it's not going to be a straight path. But you can either replace a gene that's not working right. You can inhibit a gene that's working too much. And there are a lot of gene therapy trials very soon on the horizon. So another way to find these gene therapy trials, or to find any kind of clinical trial, is to go to this website called clinicaltrials.gov and just put your diagnosis in, and you will find all kinds of different trials going on. They often focus on seizures, but it's not only seizures that the outcome will be. I think for some of these trials, there'll be cognition, sleep, all the things that are, plaguing your lives. So please do that if you haven't heard of a natural history study or some other study for your particular diagnosis. I noticed that the Rats has actually 4 separate companies working on gene therapy. So I think there's going to be more coming soon. Okay, so what happens if you don't have a genetic diagnosis? What happens if you're the patient that has the injury and it can't really be surgically approached? I think there will be gene therapies, but they're a little more complicated, but they're coming. So for instance, I'll give you an example of something that might work. So we know that certain cells in the brain are inhibitory neurons, so they sort of suppress seizures, and excitatory neurons, they cause if they're overactive, they cause seizures. So it's possible now to actually target them specifically. And you might envision a gene therapy where you go into your inhibitory cells and you make them more active, for instance. This is down the road and nobody has tried this, but I think it will be tried. So again, it's got potential to help many patients. It's just going to be a little more complicated if your child doesn't have a specific genetic diagnosis. And then on Sunday, I'm going to be talking again about RNS and DBS. The STELL trial you saw Kelly just spoke about, that was very exciting. The data was good. There was a lot of improvement in the patients, and I've been putting them in our patients for about 5 years, and I do agree there are patients that benefit a lot, and then there are some patients that don't benefit as much. But I haven't put them in very many patients, and the numbers of patients I think around the world that have been published are pretty small. So we still don't know exactly what to do in that situation, but I'll talk more about it on Sunday. So thank you. You can reach out to me via email, or it turns out we'll be having, I'll be around from 1 to 4 today if anybody wants to meet specifically. Thank you. Do we take questions? Okay. Any questions?
Yeah, if you guys have any questions for Dr. Porter, there are 2 mic runners. Just raise your hand, and we'll bring a microphone to you right up here in the front. We have one in the blue shirt. She is here, and so just remember, Dr. Porter does have time where she's spending, I think, 15 to 20 minutes with the families to talk about their cause of their epilepsy, or generally thinking, is it genetic, is it acquired, or some of these treatments she talked about. So please go to the front desk or to the LGS. This is at the registration table, I'm sorry, which is right out there by the elevators. If you'd like to sign up for her, she starts meeting with families today at 1 and is doing it from 1 to 4, and thank you for that. So we have a question here in the blue, and then there's one there in the back. We'll start in the back since the mic's back there.
Hi, so disorders like tuberous sclerosis and CDKL5 and others, they're developing targeted treatments. And what are we learning about the ability to disrupt the development of epilepsy with those targeted treatments? And is it thought, once the progression has gotten further along, what is the possibility of disrupting the LGS network through targeted treatments? In other words, are you going to have 2 treatment modalities, one for targeting the underlying genetic or other etiology causing the seizures, and then will you need a separate treatment targeting specifically the LGS network? Does that make sense?
I think so. So there is a developmental progression, obviously, to LGS. So you're not born with it, it sort of develops over time. And I guess one question is, can you reverse that? And clearly you can, to a certain extent. So I have many patients that we do epilepsy surgery on. I particularly have a tuberous sclerosis clinic, that's my specialty. And we do multiple resections now. It's quite possible to do, I think we've done as many as 8 in a patient, of focal tuber resection. And their EEG can normalize, or maybe not normalize, but they'll have some slowing based on the areas we resected, but that LGS pattern will go away. Developmentally, they respond nicely to that, like they do make developmental progress, but it's never as good as if they never had that. That's my impression. And that's the data to suggest that early surgery, for instance, so early reversal of the Lennox-Gastaut Syndrome pattern is important. That said, I do have patients who are 5 or 6 who are now talking a little bit after we did surgery at 5, for instance, who had not spoken before. So there's still some plasticity in the system, and you can fix that. Genetic treatments haven't been done, really. They've only been done really extensively in spinal muscular atrophy. So we're not there yet to know. My sense is that if we can fix the epilepsy with surgery, we might be able to fix it with gene therapy. I don't know. That's still an unknown. But earlier is better, I do believe. Yeah. Does that answer the question, sort of? Yeah, sorry. And CDKL5 is going to have some gene therapy coming along, for sure. Sorry, it's just very hard to see you, so I'm like kind of doing this weird thing.
Sorry, Dr. Porter.
That's okay.
Hello.
There you go.
Can you explain what a natural history study is?
Oh, of course, yes. So I'm running the SLC 1385 natural history study, so I spend a lot of time thinking about natural history studies. So if you think about these rare genetic diseases, we don't know a lot about them, right? We've just discovered them recently, but we don't know the full extent of how they change over time. And so a natural history study is not an intervention. It's really just collecting data so that we can understand the full spectrum of the disease. And now the FDA has said that if you have a really strong natural history study and a few number of patients, you might be able to use the natural history study so that natural progression of the disease, understanding that, to then transition into a gene therapy trial. Because let's say there's only 100 patients in the world with something, you can't have a group that's going to get no gene therapy and then compare it to a group that does. There's just not enough patients. So instead, they use this historical data that's collected through a natural history study for comparison. It also is a biomarker study, so you would be able to identify what you're going to measure once you give the gene therapy. So for instance, CDKL5 has a natural history study. Tuberous sclerosis has a natural history study. And many organizations now know that is necessary. And so they're funding these natural history studies.
Great, thank you. We just have a question from our audience at home, but before I ask it, I just want to say that I was totally wrong. It's not at the registration table. It's at the LGS Foundation table, which is across from where we had breakfast. So sorry, thank you about that. The question from home is, some with LGS, oh, wait, sorry, wrong question. Our genetic testing for our son showed an issue with SCN8A, and he seems very much like the individuals with SCN8A mutations. However, the mother or father also shows that mutation and is fine. And she's wondering if that, in conjunction with the encephalitis that her loved one suffered, could be working together to cause the LGS.
Interesting. So this is that totally situation where you have this change in the gene, and it's like, well, is this relevant or isn't it? And I don't know the specifics, obviously, in this case, but that is something to discuss with someone who is an expert in genetics, because that change in SCN8A, if the parent has it and as well, makes it much more likely that it's not disease-causing. But there are sort of machine learning or ways you can look at the genome and say, oh, it's likely to be a problem with SCN8A, like it will cause the protein to fold differently. So there's just sort of data thoughts. You can also actually get SCN8A expressed in a cell and see whether it functions normally. But that would be something to talk to a geneticist about. But it makes it much less likely if the parent has it. That's that whole de novo versus not de novo. So that would be a not de novo. And that makes us usually think that it's not causing a problem, but not always. SCN2A is one of those notorious genes where it can run in families and it can have a lot of variability. And so there are some gene changes that might be causative even if the parent has it.
Wonderful. Well, let's thank Dr. Porter again. Thank you.